Hi, everyone. Thanks, first of all, for inviting me and us to give this talk about speech biomarkers and Alzheimer's disease. I'm head of clinical researcher, and my background is I'm a neuropsychologist and a clinical neuroscientist. I'm actually located also in a memory clinic in Nice, in France, where I'm located at the moment. So I guess you might have seen those sort of figures that kind of show that neurodegenerative diseases such as Alzheimer's are, evolving over very long lengths of time, over 10 to 20 years. So you can see that even in preclinical phases where no apparent symptoms are present yet, things are changing in the brain, and it's evolving, drastically with time. But you will say that if you want to prevent or if you want to intervene, really at that stage now in Alzheimer's research that we know that we have to really tap into this early stages where I think maybe not necessarily just memory functions are impaired, but other probably more subtle cognitive changes are appearing. So how do we, I would say, call this sort of preclinical phase where early detection is most likely to have the biggest impact. And now there are even disease modifying treatment in the pipeline or even given out, for example, in the States, where it's even more really of great importance to find those people in the early stages. However, it's a big problem because our instruments and the tools we're having are maybe not sensitive enough to find those subtle changes in cognitive function. And I mean, what are our traditional dementia diagnosis tools? I mean, I'm myself working in a memory clinic. You have, you go on with a very light screener, first of all, like an MMSE, done often by a GP, or a family doctor. If he does it, it's not even safe. And then you move on to specialized tests, usually done by a neuropsychologist like me in a specific clinic. And then you go on to like more invasive testing, like brain imaging, PET, or MRI, and also you do CSF, spinal fluid tests, and blood tests also. So the early detection at now really happens in GP's office with the MMSE, or then later on. That's all we have at this stage. But as I say, when you remember the figure I showed earlier, I really would argue that the tools we're having right now in clinic are not necessarily sensitive enough. The MMSE is over 30, 40 years old, and not at all, I would say, up to date anymore. So here we come with the idea to use digital measures, since we are all day around our phone and sensors. So why not use this to detect those early symptoms? And I would say, how about using speech? So our company is a spin-off from the German Research Center of Artificial Intelligence. In 2017 we were created, and we strongly believe that speech is a window to brain health, and it can allow us to capture a lot of different changes and deficits due to different, I would say, problems on different levels in the whole process of speech production. I mean, speech is a very complex human function, and it involves a number of organs and also body parts. But as I said, if you have a deficit somewhere in these parts, these can be traced and detected through, very modern techniques and speech analysis, natural language processing. As you say, it would start always very often with the brain, where you want to produce a sentence. You have content in mind that will then also be determined your sentiment and the control of the sound. You go also through the lungs, which provides airflow to provide voice. Then there are the vocal cords that shape the sound of what you're saying, and obviously at the end also the facial muscles that modulate sound. So you can easily imagine that with all those organs and steps, there may be some diseases that are affected. And why do we believe so strongly in voice? I mean, those are the three, I would say, big arguments I can put on the table here. We can definitely measure new things. An algorithm calculates hundreds of features from a patient's speech recording, and therefore give you really new insights of a disease, gives you the opportunity to make things measurable that have been actually never looked at, because you never had something to measure it. It was maybe detected subjectively. But here we really add on this objectivity. So how is it today when you diagnose depression is basically asking your patient about depressive symptoms. The idea here would be you just talk naturally with a patient, but you can detect with speech features maybe a very typical sounding depressive voice. So you don't even have to ask it. So you can, like in the background, detect those things. And you can do it remotely. That's also a wonderful thing about speech and language analysis. You don't have to be face to face. You can use an app, even a video call, or a simple telephone. So patients don't have to travel necessarily to clinics. Remote patients can participate in trials and in research studies. There are endless possibilities to really include patients from all over the world in research around speech. So here I would say those are different disease areas where we're at the moment active in. Obviously, I will focus today mostly on Alzheimer's, where we measure cognition through speech, but we do that also in MS, for example, where same cognitive functions are affected. Motor functions like very present in ALS or Parkinson, we're trying to measure. We have also projects in COPD and asthma, as well as in psychiatry, where we try to detect, I would say, emotional affective states through speech, which is important in depression, schizophrenia, but also many other diseases. How does our speech biomarker pipeline work? We have a protocol that we usually define always with a clinical partner together, because it always depends on our target. If you want to measure cognition, obviously we're using cognitive tasks that are mainly always speech-based and record them. If you want to find out about effective emotional state, we rather would go for free and open natural speech. That is much more, I would say, likely to give emotional insight. And then, here, this is an example. We're doing verbal fluency and a verbal learning test. Also, we add some free speech. This is our protocol. We collect speech through a front end. As I said, that can be an app, that can be a phone, that can be also in clinic. We have many studies where people are just recorded in clinic. The sensor is a simple microphone where the raw input comes in. We automatically cut and prepare the audio files. For certain analysis, it has to be transcribed. For this, you can use automatic transcription or manual transcription. And then you have feature extraction, so either acoustic or also semantic features. Then we calculate a biomarker. I give you more example, a concrete example how we do that now for cognition. And then, basically it's a ready-to-use speech biomarker, either with a cutoff that can inform about certain stages, or, we can also try to predict a disease stage like MCI, etc. This can really help with accelerating those clinical trials, which are also at the moment running at the pre-screening, where we really want to identify the right person that is very likely to develop Alzheimer's disease through speech. So here you see a little bit what we are extracting. That's a little overview. Like I said, I'm talking about hundreds of features, but you can basically divide it in two layers. There's one point really to look at what people were saying. As I said, then it's really important to have a transcript. And then on the transcript, you run analysis for linguistic features, such as word frequency. Is it frequent word use or not? Syntax, the complexity of your syntax. But you also can look at semantic features like language coherence or sentiment. That is if you are using a lot of positive and negative words within your thing. It's endless, long sentences, short sentences. Are you making errors, your repetitions, etc., etc. But you can also look down there on the layer of features that describe how people are talk. That's really basically voice or waveforms. We call that acoustic features. That's like your pitch variability, how fast you're speaking, your fundamental frequency, the tone of your voice, etc., etc. If you're a jitter or shitter, if you have a strong voice, if you have a voice that breaks apart, etc., etc. Like I said, the task is very important. You have to pick. You can go from high to low cognitive load. Like I said, for emotional detection, I would definitely not make it a cognitive task. But if you want to measure cognition, we definitely want to induce some cognitive load or cognitive effort in the task, because otherwise it's too easy and just let somebody speak about their day to detect early signs of Alzheimer's, we found out is definitely not hard enough. So it's good to stick to some like norm tests that are usually already done, but then maybe extract additional features from the speech recordings. So you have picture description, for example, verbal fluency. I will talk about this as one of our tasks we worked a lot on, word list, et cetera, et cetera. Like I said, we have two front ends. We worked a lot at the beginning at studies where patients were recorded in clinic, but then moved on when the COVID happened. A lot of studies stopped, but we had our phone-based assessment already set up, and that was so convenient because that was one of the only studies that could continue to run because it was phone-based, and nobody thought at this initially at the beginning that something like COVID would happen. But we have also an app. So there is also now the possibility to do a self-administered recording protocol. There are definitely different possibilities we offer according also to the study design and also what the patient group is. If patients are too, I would say, not tech savvy, we would not go for an app, but more like the phone. Or like I said, it can also always be the simple recording in the clinic. We are active right now in 6 languages out of the box, but we are also now working on extending this into other languages. But on the left, you see all the language we are right now running studies and working in. And those again are the different domains we are tackling in, and I would definitely focus now on the cognition part because that's, I would say, the most important for Alzheimer's disease. So what we did here is we're measuring cognition through recording of two very important, I would say, very, very common cognitive tests you do in clinics, the semantic verbal fluency and the verbal learning test. Semantic verbal fluency is tell me as many animals you can think of in 1 minute. Or an item that belongs to one category. So instead of just counting the amount of animals correctly said, what is usually done, we can look at the clustering and switching of the words semantically related and the production strategy in general through the temporal aspects. How fast am I saying words together? Then you do a break. Often it's when you look for another semantic category. And on the verbal learning test, that's a very simple, test where you have 16 or 10 or 15 words heard and you have to remember them. And then you have 3 recall sessions where you always 3 to 4 recalls and then a delayed recall where you have to learn usually over time those words. So you can look at learning slopes, you can look at the arrows and also the primacy and recency effects you have in those tests. All those things you don't really usually do in the classical scoring scenario. So here, for example, you have a visualization of a semantic verbal fluency task. The person was saying cat, mouse, dog, rat, beaver. And then you can see like there's a break. And usually that's where a new semantic cluster opens. That's an executive functioning, I would say, moment. So instead of, again, just having one score, we have the whole transcript. You can see the temporal information. You can see, so those clustering and switching, we call that when cat, dog, mouse is one cluster. Obviously, they're kind of semantically related. And then up there you have also the automatic measure of word frequency. So that is if this is a word that is very highly frequent in one given language, like cat and dog, highly frequent compared to leopard or cheetah, for example. Yeah, so instead of just one score, again, you get many more scores. So in total now, we all combine that into one composite score, and that composite score is based on 85 features from both tasks. So 55 come from the semantic verbal fluency and 30 from the verbal learning test. So you have one composite score now. Or you can also break it down into 3 subdomain scores of cognition, mainly executive functioning. As I said, there's this clustering and switching that is very highly executive. And then you have also memory component, obviously, in there, and then processing speed. But all temporal aspects, how fast am I answering to those tests, is related to processing speed. So this has been anchored with other measures. I'll show you that later. But that's how we came up with this speech biomarker for cognition. So again, you can see it now. Up there, there's a global score that can be broken down into 3 subdomain scores. which has been anchored by other tests that we looked at in, like some of the trail making, for example, it's a processing speed test. We're highly correlating with our features that are related to how fast you're saying things, as I said. Then you have the executive features that were also correlated with Stroop, that's an executive function test, and the same for the memory part. And so I would say that sums up, instead of doing an hour and a half or 2 hour long neuropsych assessment, you do something that is around 10. I would say now we're around 10-minute phone-based automated assessment, so which could be very nicely done upfront as frontline screening before people come into the clinic. Not saying that the long assessment is not necessarily important to be done at one point, but maybe this can be done upfront before the person comes in. So here are some validation data. I show we have 3 cohorts in where we, really validated the speech biomarker. A Dutch cohort, an age 70. It's a birth cohort in Sweden, and Speak was a study in the UK. So we have also 3 languages here, and different patient groups as well. And you can see on the left side now the distribution of the speech biomarker score. In the Dutch cohort. The Dutch cohort was really, we had subjective memory complainers, MCI, and not that many severe, but it was a longitudinal study that is ongoing here. And compared to MMSE, which is a global cognitive test, and the CDR sum of boxes, which is another, I would say, assessment tool, you can see that our distribution is very nicely kind of showing some, there's always a bit overlap, but you see also the overlap a lot in the other 2 tools. But I would say we're kind of quite satisfied with this result. It shows that we can a little bit, I show other figures after, distinguish between those groups just based on the speech biomarkers. Here, even better. This is the age 70 cohort with over, I think, 600 participants in there. And it's a birth cohort. So most of those people are healthy and normal. There are not that many with cognitive decline. So you understand completely why there's a lot of overlap in MSE and CDR because the groups are very similar, but we see on the left side in our speech biomarker a much, I would say, stronger differentiation. So to say we might be more sensitive with our speech biomarker to distinguish between healthy controls and MCI patients. Here again, another visualization between those 3 groups in the Dutch cohort again. Just based on the speech biomarker. And here we correlated with other anchor measures like the MoCA, which I consider is a bit of a better test than the MMSE. It's not that good either, but anyways, we found a decent correlation here. But even also better, the PAC-5 is really a measurement that is used in clinical trials. And it's very highly sensitive to early decline and early stage AD. So we have a very strong correlation with our speech biomarker here in the age 70 cohort. And this is a very new, I would say, research area. Now we want to also look at biomarkers, so CSF measures like the level of tau and amyloid in the brain's fluid. And we have a lot of studies right now ongoing where we're going to look at this, because this is really what you need to get an Alzheimer diagnosis. And here we see with p-tau, we have, but it's a very small sample size. We have a small correlation here, but it's also, like I said, I think it's 35 people we have here just included. But just to say that we are looking into this right now to really compare to those, I would say, gold standard biological biomarkers. We also, of course, looked at usability. I want to say one word about this, that maybe, an automated phone call assessment is not necessarily something people are crazy about and really happy about it, but they accept it. So acceptance rate is okay. There's a trade-off. It's always nicer to be face-to-face to a person, but a lot of people are also very stressed when they see a neuropsychologist in a clinic to get tested. So the fact of staying at home and being in your own environment is definitely perceived as, an advantage. If you give them, if you give the participants the question if they prefer face-to-face or, a phone assessment, I would say the tendency goes towards face-to-face. However, we have very low dropouts and the people are really undergoing, like I said, it's 10, 15 minutes long. And acceptance rate is definitely okay. And before I stop, there's also, of course, 2 slides I want to show on the emotion motivation part, because this is still important also in Alzheimer's disease. And it's sometimes, I would say, not enough looked at. So we looked here in one study on apathy. So apathy is one of the very frequent neuropsychiatric symptoms that comes along very early already. And it's often misdiagnosed as depression, but it's a lack of motivation. It's a lack of emotional, I would say, reactivity, and a lack of goal-oriented behavior. And we looked here just in acoustic features, and we asked people to tell us about a positive and a negative thing of their life. It was a very short, from 1 minute, question. And you can see that some of the speech features correlated with the subdomains of apathy. So I don't want to go too much into detail, but it's just like to say in a more open question, and those speech features we found, for example, everything related to speech rate. That's again how fast people are speaking, or also how much. Basically, really, people with apathy didn't produce a lot of words. They had very short responses to those questions, in both questions, positive as negative. And here to see that, so you have control. Those were MC patients without apathy. They said, I would say a little bit less words in the negative, and the green is the positive story. And in apathy patients, they didn't say a lot of words in both questions. But on the right-hand side, there's a very interesting thing when you look at sentiment. That's how positive or negative I talk in one sentence, how many positive or negative words I'm using. So the balance, you can also give a score to plus and -1. So patients without apathy in a positive story talk about positive things, and a negative about negative things. And with apathy, those patients, they were really using very neutral words without a lot of emotional, I would say, load in the words. So the balance was quite neutral. And that was a very interesting result, because that's really the core symptom, what defines apathy. People are really lacking any emotional reactivity. They're not happy, they're not sad. It's blunted. And the last slide is here showing that if you combine positive, negative storytelling with our semantic verbal fluency analysis. So there's a big problem today to disentangle if somebody's depressed, a big late life depression versus the beginning of dementia. Often those diagnoses get mixed up, and patients get sent back and forth between psychiatry and memory clinic, and then they'll get treated with antidepressants, and then that's not working. And then at one point it turns out it's actually dementia. So here we could definitely see that when you look at the positive negative storytelling, patients with with depression were using much more words in the negative story than in the positive. So that's not a surprising example. That's a core of depression. People are definitely biased in negativity. To give them a negative stimuli, they will talk a lot about negative events. And the MCI and the controls were acting quite similar in their response, in their speech rate, and their number of words. And the semantic verbal fluency shows that depression patients and controls were producing the same way, animals in that fluency. However, the person with cognitive impairment, so MCI patients, were much lower and showed the different results. So we can argue, if you combine those 2 tasks together, you could probably find ways of differential diagnosis and to disentangle those 2, very often comorbid, I would say, disorders. And that's it from my side. Thank you for your attention. I hope I stayed within the time.
Yeah, thank you, Alexandra. I have just really a short question, because we always look at the progression of Alzheimer, and cognition comes very late. Are you able to detect those changes that are early, that are so subtle, that are not subjectively or objectively, with the methods that we are using right now, detectable?
I mean, the problem here is that our gold standard remains always those tests. So if we want to say we want to beat the gold standard, we have to find new gold standards. Because even still now, that's why we're looking into, for example, amyloid positivity and like other gold, or MRI, like other biomarkers that are maybe, but even that earlier affected, because I'm convinced that the neuropsych tests you do are not, they are not sensitive enough for the subtle changes, but they are our gold standard. They're our ground truth. We're all comparing to this or to diagnosis. So
but do you have longitudinal studies?
Yes, yes.
Yeah, so
we have longitudinal studies right now running where we're going to also look at, time to conversion and decliners versus non-decliners, looking who is declining in the cognitive tests and can we predict this. So this is definitely one of our main priority to do right now.
Okay. Thank you.
Lavinia,
yeah.
I have a question, if I can. Thank you so much, Alexandra. Very interesting talk and tools. My question is concerning the other sort of pathology that you have in your inclusion of MCI, because as I explained just before, it's very difficult when you have MCI to know if this patient will go in an AD biological definition or not. And in particular, as an aspect of neurodegeneration. So could you correlate your speech evaluation with the degeneration in different regions of the brain with the MRI structural functional, something like that? Or could you imagine, because I imagine if you have degeneration in a region where the language is very important, of course, the input is completely different, No? I don't know.
Yeah, no, but I agree. And again, I wish I could show now some nice fancy slides now with MRI data and structural MRI, but this really, I have a PhD student now who's going to work on this because we're running this longitudinal study with MRI where we're going to have a bit of a signature, an AD signature that defines, and then we're going to correlate our speech features with that. But also, obviously, we can see how a voice sounds of somebody who is not necessarily AD per, MRI or a biomarker, and that's of interest as well, as much as to know how AD will be reflected in the speech feature. So yeah.
But at the biological level, what do you think? It's at the biological level, the speech disorder are initiated by what? By dysfunction of the brain, aggregation of the protein in some region of the brain, or what is it's your hypothesis?
Yeah, I mean, okay. I would definitely say that it's of cognitive nature. It's the whole concept behind cognitive testing. Just that I think we push it a bit further. We go into more fine-grained analysis and variables that you can extract from a cognitive task being recorded. But yeah, I would say.
I'm sorry, we're really have to respect the time.
Thank you.
I'm sure that
thank you.
People can write to you.
Yes, exactly. Everybody can reach out. Would be my pleasure.